Hello everyone, I'm back with another helpful video just for you. Today I will be reading from class 12 biology unit 4 chapter 1. If you have watched my previous video on class 11 biology, thank you so much for the support. I'm just getting started on this journey and I'll continue posting clear and focused reading videos to make your studies easier. Let's learn and grow together. Thank you. Chapter 1 Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriad of flowers that we enjoy gazing at, the scent and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction? Flowers do not exist only for us to be used for our own selfishness. All flowering plants shows sexual reproduction. A look at the diversity of structure of the inflorescence. Flowers and floral parts shows an amazing range of adaptations to ensure formation of the end product of sexual reproduction, the fruit and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants. Flowers, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are object of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, and mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the name of 5 more flowers that are used in social and cultural celebration in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In earlier classes, you have read the various parts of a flower. Figure 1.1 will help you recall the part of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated, which leads to differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and gynoecium, differentiate and develop. You would recollect that the androecium consists of a whole of stamens representing the male reproductive organ and the gynoecium represent the female reproductive organ. Stamen, microsporangium and pollen grain. Figure 1.2a shows the 2 parts of a typical stamen, the long and slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamen are variable in flower of different species. If you were to collect a stamen, each from 10 flower, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed with each lobe having 2 theca, that is, they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various type of tissue and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a 4-sided tetragonal structure consisting of 4 microsporangia located at the corner, 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangia. In a transverse section, a typical microsporangia appears near circular in outline. It is generally surrounded by 4 wall layers. Figure 1.3b. The epidermis, endothecium, middle layer, and the tapetum. The outer three wall layer perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleated? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupy the center of each microsporangium. Microsporogenesis. As the anther develops, the cell of the sporogenous tissue undergoes meiotic division to form microspore tetrad. What would be the ploidy of the cell of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspore of pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represent the male gametophyte. If you touch the open anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your fingers. Sprinkle these grains on the drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, size, shape, colors, designs seen on the pollen grain from different species. Pollen grains are generally spherical, measuring and 25 to 50 micrometers in diameter. It has prominent two-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperature and strong acid like alkali. No enzyme that degrades sporopollenin is so far known. Pollen grains exine has prominent aperture called germ pore, where sporopollenin is absent. Pollen grains are well preserved as fossil because of the presence of sporopollenin. The exine exhibit a fascinating array of pattern and design. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. In the thin and continuous layer made up of cellulose and pectin, the cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant food reserved and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grain are shaped at this two-cell stage. In the remaining species, the generative cell divide mitotically to give rise to the two male gametes before pollen grains are shed, that are three-cell stage. Pollen grains of many species cause several allergic and bronchial affections in some peoples, often leading to chronic respiratory disorder like asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with import wheat has become un It may be mentioned that Parthenium or carrot grass that came India as the contaminant with import wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grain are rich in nutrients. It became a fashion in recent years to use pollen tablet as food supplements. In Western countries, a large number of pollen products in the form of tablet and syrup are available in the market. Pollen consumption has been claimed to increase the presence of athletes and race horses. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen sperms of many animals, including human, or for artificial insemination. It is possible to store pollen grain of a large number of species for years in liquid nitrogen, that to minus 196 degrees Celsius. Such stored pollens can be used as pollen banks similar to seed bank in crop breeding programs. The pistil, Megasporangium, and embryo sac. The pistil, Megasporangium, and embryo sac. The gynoecium represent the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocapillary, or may have more than one pistil, multicapillary. When there are more than one, the pistil may be fused together, that is syncarpous, or may be free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serve as a landing platform for pollen grain. The style is the elongated, slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11. Arising from the placentas are the megasporangia, commonly called ovules. The number of ovules in an ovary may be one, like wheat, paddy, and mango, or to many, papaya, watermelon, and orchids. The megasporangium, ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuse with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective evolves called integuments. Integument encircle the nucleus except at the tip where a small opening called the micropyle is organized. Opposite the micropylar end is the chaliza, representing the basal part of the ovule. Enclosing within the integument is a mass of cell called the nucellus. Cells of the nucellus have abundant reserve food material. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovule generally differentiate a single megaspore mother cell MMC in the micropyle region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspore, female gametophyte. In a majority of flowering plant, one of the megaspore is functional while the other 3 is degenerate. Only the functional megaspore develop into the female gametophyte, embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cell of the nucellus MMC, the functional megaspore and female gametophyte?
Let us study about the formation of the embryo sac in details. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two nucleated embryo sac. Two more sequential mitotic nuclear divisions result in the formation of the 4 nucleate and later the 8 nucleated stages of the embryo sac. It is of interest to note that this mitotic division are strictly free nuclear, that is, nuclear division, are not followed immediately by cell wall formation. After the 8 nucleated cell stage, cell wall are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac. 6 of the 8 nuclei are surrounded by cell wall and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and continue the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have special cellular thickening at the micropyle tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazial end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, the 8 nucleate is 7-celled. Pollination. In the preceding section, you have learned that the male and female gamete in flowering plant are produced in the pollen grain and embryo sac, respectively. As both type of gamete are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain straight from the anther to the stigma of the pistil is termed pollination. Flowering plants have evolved an amazing array to adaptation to achieve pollination. They make use of external agent to achieve pollination. Can you list the possible external agent? Kind of pollination. Depending on the source of pollen, pollination can be divided into 3 types. Number 1, autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grain from the anther to the stigma of the same flower. In a normal flower which open and expose the anther and the stigma, complete autogamy is rather rare. Autogamy in such flower requires synchrony in pollen release and stigma receptivity, and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis and camelina produce 2 types of flower. Chasmogamous flower which are similar to flower of other species which exposed anther and stigma, and clistogamous flower which do not open at all. In such flower, the anther and stigma lies close to each other. When anther dehiscence in the flower buds, pollen grain come in contact with the stigma to affect pollination. Thus, clistogamous flower are invariably autogamous as there is no chance of cross pollen landing on the stigma. Clistogamous flower produce assured seed set even in the absence of the pollinator. Do you think that clistogamy is advantageous or disadvantageous to the plant, and why? Ketonogamy. Transfer of pollen grain from the anther to the stigma of another flower of the same plant. Although ketonogamy is functionally cross-pollination involving a pollinating agent. Genetically, it is similar to autogamy since the pollen grain come from the same plant. Xenogamy. Transfer of pollen grain from anther to the stigma of a different plant. This is the only type of pollination which during pollination bring genetically different type of pollen grain to the stigma. Agent of pollination. Plant use two abiotic, wind and water, and one biotic animal agent to achieve pollination. Majority of plants use biotic agent for pollination. Only a small proportion of plant use abiotic agent. Pollen grain coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollen when compared to the number of ovule available for pollination. Pollination by wind is more common amongst abiotic pollination. With pollination also require that the pollen grain are light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamens so that the pollen are easily dispersed into the wind current, and large often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The ears you see are nothing but the stigma and style which wave in the wind to trap pollen grain. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plant and is limited to about 30 genera. Mostly
monocotyledons. As against this, you would recall that water is the regular mode of transport for the male gametes among the lower plant groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zostera. Not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flower emerged above the level of water and are pollinated by insect or wind, as in most of the land plant. In Vallisneria, the female flower reach the surface of water by the long stalk, and the male flower of pollen grain are released on to the surface of water. They are carried passively by water current. Some of them eventually reach the female flower and the stigma in another group of water pollinated plants such as sea grasses. Female flower remain submerged in water and the pollen grain are released inside the water. Pollen grain in many such species are long ribbon-like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grain are protected from wetting by mucilaginous covering. Both wind and water pollinated flower are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plant use a range of animal and pollinating agent: bee, butterflies, flies, beetle, wasp, ants, moth, birds, sunbirds, and hummingbirds and bats are the common pollinating agent. Among the animal insects, particularly bee, are the dominant biotic pollinating agent. Even larger animals such as some primates, lemur, arboreal, tree-dwelling, rodent, or even reptiles, peacock lizard, and garden lizard, have also been reported as pollinator in some species. Often flower of animal pollinated plant are specifically adapted for a particular species of animal. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flower by color and or fragrance. The flower pollinated by flies and beetles secrete fall odor to attract these animals. To sustain animal visit, the flower have to provide reward to the animal. Nectar and pollen grains are the usually floral reward. For harvesting the rewards from the flower, the animal visitor comes in contact with the anther and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay egg. An example is that of the tallest flower of Amorphophallus. The flower itself is about 6 feet in height. A small similar relationship exists between a species of moth and the plant yucca, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larvae of the moth come out of the egg as the seeds start developing. Why don't you observe some flower of the following plants or other available to you: cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinator. You will have to patiently observe the flower over a few days and at different time of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visited. Carefully observe if any of the visitor come in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are preferred to as pollen or nectar robber. You may or may not be able to identify the pollinators, but you still surely enjoy your effort. Outbreeding devices. Majority of flowering plants produce hermaphrodite flower, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination result in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma respectively are not synchronized. Either the pollen is released before the stigma become receptive, or stigma become receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both this device prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower to other flower of the same plant from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is to produce of unisexual flower if both male and female flower are present on the same plant, such as castor and maize monoecious. It prevents autogamy but not geitonogamy. In several species such as papaya, male and female flower are present on different plant, that is, each plant is either male or female dioecious. This condition prevents both autogamy and geitonogamy. Pollen-pistil interaction. Pollination does not guarantee the transfer of the right type of pollen-compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the other species from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or the wrong type, incompatible. If the pistil accept the pollen and promote post-pollination event that leads to fertilization, if the pollen is the wrong type, the pistil reject the pollen by preventing pollen germination on the stigma of the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by acceptance or rejection in the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical component of the pollen interaction with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognized followed by acceptance of rejection. As mentioned earlier, following compatible pollination, the pollen grain germinate on the stigma to produce a pollen tube through one of the germ pores. The content of the pollen grain move into the pollen tube. Pollen tube grow through the tissue of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grain are shed at two-celled condition, a vegetative cell and generative cells. In such plant, the generative cell divides and form the two male gamete during the growth of pollen tube in the stigma. In plant which shed pollen in the three-celled condition, pollen tube carry the two male gamete from the beginning. Pollen tube, after reaching the ovary, enter the ovule through the micropyle and then enter on the synergid through the filiform apparatus. Many recent studies have shown that the filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. All this event form pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen-pistil interaction. As pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction. Even in incompatible pollination to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam, and vinca on a grass site containing a drop of sugar solution, about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tube coming out of the pollen grain. A breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior variety. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grain are used for pollination and stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female parent bear bisexual flower, removal of anther from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contaminating of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attain respectively mature pollen grain collected from anther of the male parent are dusted on the stigma, and the flower are rebagged and the fruit allowed to develop. If female parent produce unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma become receptive, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergate, the pollen tube releases the two male gamete into the cytoplasm of the synergate. One of the male gamete moves towards the egg cells and fuse with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gametes move towards the two polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus, PEN. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two types of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomenon is termed double fertilization. An event unique of flowering plants, the central cell after triple fusion becomes the primary endosperm cell, PEC, and develops into the endosperm while the zygote develops into an embryo. Post-fertilization structure and event. Following double fertilization, event of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit are collectively termed post-fertilization events. Endosperm. Endosperm develops precedes embryo development. Why? The primary endosperm cell divides repeatedly and form a triploid endosperm tissue. The cells of the tissue are filled with reserved food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconuts that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei. And the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, bees, before seed maturation, or it may persist in the mature seeds, example castor and coconut, and be used up during seed germination. Splits open some seeds of castor, peas, beans, groundnut, fruits of coconuts, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Through the states differ greatly. The early stage of embryo development, embryogeny, are familiar in both monocotyledons and dicotyledons. Figure 1.13 depicts the stage of embryogeny in a dicotyledonous embryo. The zygote gave rise to the pre-embryo and subsequently to the globular heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule of stem tip. The cylindrical portion below the level of cotyledons is hypocotyledons that terminates at its lower end in the radicle or root tips. The root tip is covered with a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledons is called scutellum, that is situated towards one side of the embryonal axis. At its lower end, an embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaves primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnuts, overnight. Then split the seeds and observe the various parts of the embryo and the seeds.
Seed. In angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats, cotyledons, and an embryo axis. The cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserve as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example, pea groundnuts. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. Example, wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucleus nucellus are also persistent. This residual persistent nucellus is the perisperm. Integument of ovule harden as tough protective seed coat. The micropyle remain as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed mature, its water contain in residue and seed become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slow down. The embryo may enter a state of inactive called dormancy, or if favorable conditions are available, aquatic, moisture, oxygen, and suitable temperature, they germinate. As ovule mature into seeds, the ovary develop into a fruit, that is, the transformation of ovule into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develop into the wall of fruit called pericarp. The fruit may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seeds present in the fruits? In most plants, by the time the fruit develops from the ovary, other floral part degenerated and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruits are called false fruit. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruit are the result of fertilization. There are a few species in which fruit develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is such example. Parthenocarpy can be induced through the application of growth hormone and such fruits are seedless. Seeds offer several advantages to angiosperm. Firstly, since reproduction process such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitat and help the species to colonize in other areas. As they have sufficient food reserved, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being product of sexual reproduction, they generate new genetic combinations leading to variations. Seeds is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to rise crop in the next season. Can you imagine agriculture is the absence of seeds or in the presence of seeds which germinate straight away soon after formation and cannot be stored? How long do the seed remain alive after they are dispersed? This period again varies greatly. In a few species, the seed lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of Lupinus arcticus, excavated from Arctic tundra
The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year-old viable seed is of the date palm, Phenoxy dactylifera, discovered during the archaeological excavation at King Herod Palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to compare the enormous reproductive capacity of some flowering plant by asking the flowering questions: how many eggs are present in an embryo sac, how many embryo sacs are present in an ovule, how many ovules are present in an ovary, how many ovaries are present in a typical flower, how many flowers are present on a tree, and so on. Can you think of some plant in which fruit contain very large number of seeds? Orchid fruit are one such category, and each fruit contains thousands of tiny seeds. Is the case Similar is the case in fruit of some parasitic species such as orobanche and stringer. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from that tiny seeds? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the year? Apomixis and polyembryony. Although seeds in general are the product of fertilization, a few flowering plants, such as some species of Astacia and grasses, have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a formation of asexual reproduction that mimics sexual reproduction. There are several ways of develop of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develop into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac that dividing protrude into the embryo sac and develop into the embryos. In such species, each ovule contain many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different size and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clone? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrid has tremendous increased productivity. One of the problem of hybrid is that hybrid seeds have to be produced every year. If the seed collected from hybrid are shown, the plant in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seeds is costly, and hence the cost of hybrid seeds become too expensive for the farmer. If these hybrids are made into apomictic, there is no segregation of character in the hybrid progeny. Then the farmer can keep on using the hybrid seed to raise new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seeds industry, active research is going on in many laboratories around the world to understand the genetic of apomixis and to transfer apomictic genes into hybrid varieties. Summary. Flower are the seat of sexual reproduction in angiosperm. In the flower, androecium consisting of stamen represent the male reproductive organ, and gynoecium consisting of pistil represent the female reproductive organ. A typical anther is bilobe, dithecus, and tetrasporogiate. Pollen grain develop inside the microsporangia, four-wall layer, the epidermis, endothecium, middle layer, and the tapetum surround the microsporangium. Cell of the sporogenous tissue lying in the center of the microsporangium undergo meiosis, microsporogenesis, to form tetrad of microspore. Individual microspore mature into pollen grain. Pollen grain represent the male gametophytic generation. The pollen grain have a two-layer wall, the outer exine and inner intine. The exine is made up of sporopollenin and has germ pore. Pollen grain may have two cells, a vegetative cell and generative cell, or three cells, a vegetative cell and two male gametes. At the time of shedding, the pistil has three parts: the stigma, style, and the ovary. Ovule are present in the ovary. The ovule have a stalk called funicle, protective integument, and an open called micropyle. The central tissue is the nucellus, in which the archiporium differentiated. A cell of the archiporium, the megaspore mother cell, divide meiotically, and one of the megaspore form the embryo sac, the female gametophyte. The mature embryo sac is 7 cell and 8 nucleated. At the micropylar end is the egg apparatus consisting of 2 synergid and an egg cell. At the chalazal end are 3 antipodal. At the center is the large central cell with 2 pollen nuclei. Pollination is the mechanism to transfer pollen grain from the anther to the stigma. Pollination agent are either abiotic, wind and water, or biotic, animal. Pollen pistil interaction involve all events from the landing of pollen grain on the stigma until the pollen tube entered the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grain germinate on the stigma and the resulting pollen tube grow through the style, enter the ovule, and finally discharge two male gamete in one of the synergid. Angiosperm exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The product of this fusion are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develop into the embryo and the primary endosperm cell form the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular, and heart-shaped stage. Before maturation, dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledons. After fertilization, ovary develop into fruit and ovule develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomictic have several advantages in horticulture and agriculture. Some angiosperm produce more than one embryo in their seeds. This phenomenon is called polyembryony.
Thank you so much for watching. If you enjoyed this video, please do not forget to like, share and subscribe to my channel. Also hit the bell icon so you never miss any updates. See you in the next video. Thank you.